Hi, we know the chemical differences between DNA and RNA. The primary difference is that RNA has ribose, whereas DNA has deoxyribose as sugar. There are also differences in terms of nitrogenous bases. RNA has uracil while DNA has thymine, and chemically these two differ just by one methyl group. Thymine has an extra methyl group on the 5th carbon of the nitrogenous base. So why should there be this kind of difference, or what is the evolutionary significance of it? This has connection with water. Somehow where water is, there is dynamicity, there are reactions, and that is how water makes life possible. We are all set of networking reactions walking around. Let's try to see what the connections are. I'm Dr. BCMR, Assistant Professor at Shastra. There is something about water that it can make reactions happen, and then so it is a continuous way of that is how it keeps the system dynamic because of its hydrogen bonding capacities and the polarity. We will consider some of the examples that are here, and then you will see. So here is a cytosine that is present, and the cytosine is a small molecule, whereas the DNA is, yes, the cytosine is present here, and cytosine has an amino group and one keto group, and there is a probability that because of the hydrolytic activity of the water, the amino group might be lost. That is called as deamination, removal of the amino group as ammonia, and that is mediated by water. And as a result, it will end up forming uracil. So cytosine upon deamination forms uracil, and that is a problem. And let's see how it happens. We will focus at this point on the 3rd cytosine that is present here. Say, for example, the 3rd cytosine that is present, it has undergone deamination, and it has formed uracil. So if uracil was a normal or natural base within DNA, how could the repair systems, or anyway, is there any other way that can distinguish uracil from that of deaminated cytosine, which is also uracil? So there is a problem. That means there should be something else to prevent or to be able to distinguish between what is uracil and what is a deaminated cytosine. And we have to also remember that inherently the nitrogenous bases do have different stabilities, and also based on oxidation and many other aspects, which we will not go into. But logically, if we look at the problem, that is there a way to keep the DNA or keep the genetic information intact? Is there a way to distinguish the normal uracil, if it were a natural base within DNA, from that of deaminated cytosine? It is not possible. So that is one biggest problem that would be, and therefore it is thought that evolution has come up with a methyl group on uracil at the 5th carbon that is nothing but thymine. And as it is believed that RNA is the first molecule that has formed out of these two, DNA and RNA, and RNA's world is what it means to say that RNA was the genetic material and it performed all the activities of propagation, and then later for stability DNA has evolved. So DNA evolution also, the 2' OH can get deprotonated, because of which an oxyanion could form and break the phosphate backbone, so the degradation of the nucleic acid could happen. That is possible with RNA, especially in conditions of where there is alkaline conditions. So DNA has deoxyribose, meaning that the 2' OH, the O is missing. So even if it is in alkaline conditions, there is no probability of an oxyanion forming, and therefore the backbone is stable. What we are presenting here, or what we are discussing here, is more about informational stability, not the structural stability. 2' OH and 2' H is about structural integrity of the genetic information. In this case, or the physical integrity. In this case, about thymine and uracil, which one should be present, it's more of an informational integrity. So that is why it is thought that DNA has, or the evolution has brought in thymine to be able to distinguish cytosine, cytosine, and now if there is an uracil within DNA, it is clear that there is one way, or the most likely way that it has formed is because of deamination of cytosine, and that is why it is thought that thymine has evolved within and then DNA. Similarly, deamination can also cause several other problems, especially this is much more prominent in cases of eukaryotes, where methylation of cytosine is common, especially in CpG islands, which are in regulatory regions of several genes, which is through epigenetic regulation. So DNA is methylated, and in that DNA, cytosine is the one that is methylated, as depicted here. That is 5-methyl cytosine. This is a very common one. Now, if we see deamination of methyl cytosine, if this methyl group is lost, what we have is the formation of thymine. And so there is normal thymine. There is normal thymine in 5th position here, and there is in 6th position we have a thymine, which actually is a deaminated 5-methyl cytosine. And now for repair mechanisms to distinguish between which one to remove is a difficulty. Of course, there is a possibility that we have a TG mismatch and mispairing, or you can say it as a mismatch, and then there are repair mechanisms which can remove the thymine in those cases. Nevertheless, there is a possibility, if replication did occur in these scenarios, then it is going to be established as a mutation. Water can also do many other things. That is, it can even break the glycosidic bond that is present here, and that could result in the loss of whole base from that DNA, and that is, it will be left with an epimeric site. So the whole base is lost. So water, through its activity of and how it can play this dynamicity, can have an effect on the genetic information. Of course, there are dedicated repair mechanisms to try to undo the damage, but still there will always be leakage of some, and then there will be establishment of mutations. This is much more the one with 5-methyl cytosine and the formation of thymine is a very good or hot spot for mutagenesis, because the probability of the thymine skipping or being counted as legitimate base will cause a lot of problems, or mutagenesis will establish the formation of mutations. We'll see you again in the next. Thank you for watching. Have fun.